What's up, everyone? How are you? I hope you're doing well. Today, we're talking about DNA, and we've looked at DNA, the structure, the primary, secondary, tertiary, and quaternary structures. We know what DNA does and all that stuff. Today, I'm talking about some of the methods that we use in the lab, in research, in industry to manipulate DNA in terms of studying DNA, creating new DNA, creating mutations, cloning, and things like that. So I'm going to look at a few methods. Some are very old, some are very new, but these are a few methods that we use to study DNA. So let's jump into it. So, recombinant DNA, cloning, chimeric genes, and synthetic biology. So, we'll talk about that and more. Let's start by cloning. Now, cloning DNA goes back a long time ago, and we can make now any type of DNA clone that you want. So, a clone really refers to a collection of molecules or cells all identical to the original molecule or cell. Now, the clones usually happen naturally through cell division, but we found a way to use that machinery, that natural machinery, to do this in the lab. So you can clone any gene you want and you can make the exact copy of that gene. You can also make an altered version of it to study a certain mutation. So we call it recombinant DNA technology. What do we use to do that? We use plasmids. We use E. coli and bacteria. And these are really good because you can insert into a E. coli plasmid, you can insert any gene and it's going to make it for you. And it's going to not only multiply it, but it will make the protein from that gene. It's really cool. So we use E. coli a lot, and E. coli has a plasmid. Now, bacteria DNA is very interesting in that instead of being a double helix, it's just a circular double stranded DNA that we call plasmids. And these plasmids can be cleaved by restriction enzymes. Right? And when you cleave by restriction enzymes, leaving these sticky ends, you can bring in the protein, not protein, but a gene and put it in the insert. Right? And that's what we call recombinant genes or recombinant molecules. So let me show you an example. So this is how we do it. We have a circular DNA, which is a plasmid. You have a gene of interest, and then you cut this with a restriction enzyme. EcoR1 is one of the most famous restriction enzymes. So you cut it, you insert it in there, and then you use DNA ligase to ligate the protein. To ligate to the DNA, and look, you have a new plasmid that has your DNA in it. So we call this recombinant plasmid or just chimeric plasmids. So chimeric DNA, that's what we're talking about. All right. So when these plasmids, right, are designed using vectors. So the vectors, these cloning vectors are these plasmids that you can modify. So this is a famous template of a vector, and it's going to have a gene for ampicillin resistance. It also has a gene for many other things, but it has the origin of replication. Very important, right? It has an origin of replication. It has antibiotic resistance, and then it also has a cloning site. So you add all of this, you add your protein into this vector, and it's going to make it. Now we have companies. For example, when I make my protein, when I make my DNA, I do this on a computer. I submit to a company, and they send me an already made, ready to go plasmid, right? but this is a vector, and you can design this and put in anything you want in there, and the bacteria is going to make it. It's really, really cool. The ampicillin resistant gene, by the way, is important because bacteria is everywhere, and when you're growing bacteria, it's not just the bacteria that you want that's going to grow. Everything is going to grow. But we know that ampicillin kills bacteria. So when you have your ampicillin resistant gene in this plasmid, the plasmid that you're trying to isolate will not die. It's a beautiful way to isolate this plasmid that has your gene of interest. So that is plasmid DNA. Another thing that you must know when it comes to this, we use this system of the bacteria called lac operon, right? And lac operon is very intricate machinery where you can not only synthesize your gene of interest, but you can also control the synthesis of it, where you have a replacer, you have an inducer, right? So that, for example, when you're ready for the bacteria to make your protein, you add in an inducer there, right? And the inducer is going to start at the origin of the replication, right? So you have usually the origin of replication is repressed by a repressor, right? But when you bring in an inducer. So in this case, for lac operon, we add lactose. Lactose binds to the replacer. Replacer is released. And then when it's released, the DNA polymerase can come and go to the promoter, and then boom, we make the enzymes that we want to make. And you can come in here and put in another gene in here, and it's going to make it too. So it's not just these. All right. So this is a good machinery that we use to control this DNA production. And we use all these methods to make a DNA library. What's a DNA library? A DNA library of a certain cell or a certain organism is going to be a set of cloned DNA fragments that together represent the genes of a particular organisms, right? So these libraries used to be very useful, especially if you don't have the full clone. So let's say we want to look at the DNA sequence of an amoeba, for example, and we don't have the full profile of the DNA sequence, but we know that these 25 genes are found in an amoeba. What do? You have a fragment that contains all those genes that we know are in an amoeba, and this is a DNA library for an amoeba. All right, so that's what we use DNA libraries for. And how do we make some of the DNA library? How do we use them? We use them to screen for non-DNA segments. So let's say, and we call this colony hybridization. So this is a way to screen plasmid-based genome libraries for a DNA fragment of interest, right? So if you have a host bacteria that contains the plasmid-based library of DNA fragments, you're going to put them in a petri dish. You make a replica of them using a disk, and then that disk that you made, right, you can then find, so you can hybridize that with your sample of interest to see what binds to that disk. So I think I have an example here, right? So you see this one. You have your normal bacterial colonies, right? And then you put them in a disk. And then after you take the disk, you take your sample of interest, right? This is a sample of interest, and this sample of interest will have fluorescent material, right? So you use a labeled probe. Why is this important? It's because you're going to add this into your disk, and guess what? Only the DNA that can bind to this is going to pretty much stay. And then after you wash off everything, you find that, for example, in this case, only two colonies from your original sample contain the DNA that you want that was on the disk. So this is a way to, we call this colony hybridization, and it's a way to see like, hey, in this library of colonies that I have, is this DNA that I have corresponding to any of these colonies, or does it belong to any of these colonies? So that's colony hybridization. And on top of DNA library, there are what we call cDNA libraries. cDNA libraries. So cDNA is better than DNA. Why? Because we talked about how mRNA is more representative of what's happening in the cell compared to DNA. Because not all the DNA is expressed, right? Not all DNA is expressed. But mRNA shows gene expression. So you can, and we know how to turn, to copy mRNA into DNA, right? So you use the mRNA and then you copy it into DNA. We call this cDNA, right? cDNA is DNA that is constructed from mRNA. So the cDNA libraries will be constructed by synthesizing cDNA from purified cell mRNA. And these are really good because they are more representative of what's happening. So cDNA library are better than just pure DNA library, all right? And we make them, so this is an example of how we make them, where you start with so this is an example of how we isolate mRNA from the cell, right? But we won't talk too much about it, but this is just, we use column chromatography, and we use different concentrations of salt in order to separate the mRNA. And after you separate the mRNA, you form cDNA libraries on it. Now, let me ask you this. You might have heard of this. What do we use to turn DNA, to turn mRNA to cDNA? What do we use? We use this enzyme called reverse transcriptase, right? Reverse transcriptase is an enzyme that synthesizes DNA strand from mRNA. So viruses use this as well. So how do we do that? mRNA, right? So this is how reverse transcriptase synthesizes cDNA. So you're starting with an mRNA. All right. And then you synthesize the first strand. And then you have elongation here. All right. And then here you can even add DNA polymerase. So it's not just reverse transcriptase. DNA polymerase comes in and removes any fragments that have been made here. So you have not only reverse transcriptase, but you also have polymerase, right? So this is, by the way, this is because you have different fragments depending on which genes you have, and you have different primers as well. All right. So after you have your cDNA, guess what? You can actually use different different enzymes, not different restriction endonucleases, to cut it so that you can insert it into the bacteria. All right. So that's enough on cDNAs. Now let's talk about PCR. PCR is this life-changing method that pretty much changed DNA research completely. Why? Because with PCR, you can make billions and billions of just one copy of a gene, and it's very, very cheap, very good, very efficient. How does it do it? You're going to open up the DNA, right? So you have your double strand here. You open up the DNA so that you open your strands, and then you bring in DNA polymerase and then single nucleotides so that you can make complementary strands, and then you repeat. How do we do that? Temperature. That's why. And the guy who invented PCR, was, there's a whole story about him, but he was an outside the box thinker, to say the least. But he said that he was just, I think, surfing when he thought of this. But it's a very, very genius way to do this, because you bring up the temperature, the strands separate, you bring down this temperature, they make they use the nucleotides to make another copy, and then you bring up the temperature again, they separate, you bring down the temperature, they get together, and over again, and you make a lot of copies. So we can use this, and usually a typical PCR takes about an hour and a half, and you have so many cycles. So this is 3 cycles, but you can think about it. We use a lot of cycles, and we come up with a lot of DNA. We use this nice little machine. This is called a thermocycler. So you put your tubes in here into the thermocycler, and then you close it. You see this top right here? This is the one that heats up. So it goes directly above your tubes, and then it can heat up and then go down. That's why we call it a thermocycler. So this is an example of your cycle, where it's going to go up, go down, then go up again, like that, right? This is an example of a cycle that goes up over and over again. But you can, in this machine, you can set it up and actually set up the temperatures that you want. It's really, really neat. And if you don't get enough DNA, you can optimize it. But that's what PCR is really. You change the temperature so that you can open up the strands, and then you can multiply your DNA. Now, I won't talk too much about it, but one thing to note is that usually, I think we call it a master mix when we do it, but you have to have enzymes, primers, and nucleotides and your DNA into a solution, right? And these nucleotides, as a reminder, these are triphosphate nucleotides, right? So DATP, DTTP, DGTP, DCTP, right? So you add the nucleotides, you add the polymerase, you add the primers, you add the DNA. There are other things that you add to help visualize your DNA on a gel, for example, if you want to run it on a gel. But that's the basic of PCR. Now, let me talk about a special type of PCR called RT-PCR. RT-PCR is PCR, but it has RT in it. What's RT? Reverse transcriptase PCR, right? So it's a variation of PCR where instead of looking at the DNA, instead of multiplying the DNA, we're multiplying it from the mRNA. So again, this is more accurate because mRNA is more representative of what's happening in the body or in the cell. So we use reverse transcriptase to synthesize the DNA, and then after that we do the PCR. We multiply the DNA that came from the RNA, reverse transcriptase RNA. So here is that's where we use cDNA, right? The cDNA is the copy of the DNA that came from the mRNA. So that's the one that we use. So this is kind of a quick overview of the method where you come up with a sample, right? You isolate RNA from the sample, and then you use reverse transcriptase to go from RNA to cDNA. And then when you have the cDNA, guess what you do? You do PCR, right? And the PCR is exactly the one we talked about, where you add the primer and all that stuff so that you can come up with a lot of copies of this DNA. This is RT-PCR, right? And we also have a nice machine that can do all of this, a machine for RT-PCR. RT-PCR was very, very useful in the COVID era because this is the one that we used to detect whether or not somebody has a coronavirus. So when they say PCR test, it wasn't a PCR test. It was RT-PCR. And those are two different ones. They were not looking at the DNA. They were looking at the mRNA, the expression of a particular viral protein. That's what the PCR test was. Now, RT-PCR is also called, instead of reverse transcriptase, is also called real-time PCR because you can look at it in real time, meaning, looking at the expression. You also have something called RT-qPCR. So RT-qPCR is quantitative PCR. So instead of just measuring the presence of something, it also measures the quantity, right? So you're going to look at the quantity or the level of expression, and it's really, really good. And qPCR usually looks like this depending on how many PCR cycles. So you can compare not only whether a gene is expressed, but you can also measure how much compared to another person or another organ or another cell, right? So that's RT-qPCR. You have other sophisticated methods such as microarrays. Microarrays are also ways to measure DNA. And how do we measure them? We have DNA libraries and you immobilize them on a chip, right? So these are arrays of DNA, different oligonucleotides immobilized on a chip. So if you have a chip, right, and then you have something immobilized on it, you have libraries that are immobilized on it, right? What you can do is you can add your DNA, right? You add your DNA, the DNA that you want to study, and then you go through different cycles and different things, but what you end up with is find matches, right? You try to find the matches, to find the level of expression. And we use microarrays to see something. For example, you can look at a cancer cell versus a normal cell and see which genes are expressed versus which genes are repressed. We use this a lot and you'll see something that looks like this is a microarray that say that, okay, for example, the result you're gonna see is gonna be like, okay, this is the yellow. The yellow here means that you have red and green. So green is only expressed in cancer cells, in normal cells. Red only expressed in cancer cells, and then yellow expressed in both. So you get a result like this. And again, it comes from a microarray. This one contains like 12, but it's going to contain hundreds and hundreds of genes. So you can study the expression of so many genes in a particular cell using microarrays. All right, protein expression, we talked too much about it, but you can use the machinery, the machinery that we talked about to express a lot of proteins. So all this DNA that we just talked about, you can use the bacterial machinery to synthesize protein from all of these DNAs, right? Oh yeah, this is an example of a microarray. This is a real microarray, and you can see how you have an expression level that shows you the green means is expressed in wild type. The red is expressed in disease. You're going to have where there's no expression at all. You're gonna see where you have less green or more yellow and things like that. All right, I was just talking about protein expression. One thing that I've been trying to do in my research is not only to express your protein in E. coli, but express it in mammalian cell cultures. And that's a little bit harder, but we do that as well, where you can just clone a certain protein or a mutation, and then you can put it in your mammalian cell culture so that you can express it as well. But there's a whole area and a whole group of people trying to make this easier. All right, so if you have a DNA library, can you express it? Yes, we can. So going back to the vector that I was talking about, you can use the vector and look at the vector. The vector has LAC-Z and all that other stuff. This is an operon. We're talking about the operon, the LAC operon thing, right? So you can use this machinery and this vector to express your protein. All right, and these are some of the examples of proteins that have been isolated. They were isolated early, very early, using the clone genes in the libraries. But right now, we can pretty much express anything. Right, there's some things that we call reporter genes. Reporter genes are really good in that they are genes that we use to measure the expression of another gene. So we talked about GFP, green fluorescence protein, and this is a reporter gene that you can take and you can insert it in front of a gene. So that's how this is what you do. You take a reporter gene, you insert it in the front of a gene that you want to express, so that whenever that gene gets expressed, the reporter gene also gets expressed, and then you can measure the expression of a reporter gene. Why do we do that? Why can we not measure the expression? Some genes are easier to measure the expression of. For example, the GFP gene is a green fluorescence gene, right? If fluorescence is green, so we can use that as a reporter gene, so that whenever your gene gets expressed, the GFP gets expressed as well, and you can measure the expression of any gene because of that. So we use this a lot. Right, but a reporter gene are chimeric DNA molecules composed of gene regulatory sequences positioned next to an easily expressible gene product. Right, emphasize that. So GFP is one of the biggest report right genes that we use, but we use other genes as well. Another thing that you can use or do using a reporter gene is protein expression, right? Mainly protein interaction that involve DNA. So here is an example of there is no expression if there is no interaction between these two proteins, but whenever these two proteins interact, there is expression, and the reporter gene here is expressed. So you see signal. You only see signal when there is interaction. And what's not shown here is also that we use reporter genes to look at proteins that bind to DNA, right? Proteins that bind to DNA. You can use that as well. All right. Immunoprecipitation. This is a really, really common method. You probably have heard of this. And the immunoprecipitation, whenever you hear immuno, when it comes to these techniques, antibodies. That's pretty much what you gotta hear. Immuno, antibodies. So we use antibodies. And then precipitation, you know what precipitation is. Precipitating something out of the solution so that you can centrifuge it down. So this is what happens. We use immunoprecipitation to see which proteins, bind to which proteins. So we're looking for protein complexes or protein-protein interaction. So what do? Let's say you have two different proteins here, right? And you want to see whether or not they are together, right? We want to see which proteins get together. Well, first of all, you add antibody. So this is the antibody. You see the antibody here? So if you add the antibody for a particular protein, so we added the antibody for the red protein here. So we really want to see like which protein is in a complex. So antibody for the red protein. And then after antibody, you add what? You add, you're going to add the beads, right? Beads or anything that's going to bind to that antibody, right? So this is the precipitation step. You're going to find something that's going to go and bind to that antibody so that it can precipitate it out of the solution, right? And then after you added the beads that are going to add to bind to the antibody, you centrifuge. You centrifuge so that everything that's bound to the antibodies go down, right? So after this, you're going to analyze what's precipitated. And remember, you targeted one protein, right? You only targeted one protein with your antibody. So the idea is that, okay, let me analyze what precipitated and see if there is another protein that came out with this other protein that I targeted. So this is a way to find the complex because, for example, here, look at the one at the bottom. The red has the green protein bound to it. So the green protein also precipitated, even though you wanted to precipitate the red, you targeted precipitation of the red protein, but the green protein also precipitated because it's bound to it. And you can measure this, right? So this is a really, really easy way to measure protein complex or protein-protein interaction. And in case of my research, I add a drug that's known to inhibit this interaction, and then I see how the immunoprecipitation is affected, right? If I don't see anything at the bottom here, for example, if I only see red and not green, I'm like, oh, okay, my drug inhibits the formation of this complex, right? So co-immunoprecipitation is very, very important. All right. These techniques are some of my favorites, especially Western blot. I did probably what maybe I would say 500 Western blots because I did most of them when I worked at UAMS. But you have Southern, Western, and Northern blotting, and they are all related. So what are they? Let's start with Southern blot. So Southern blot has to do with DNA. So by the way, yeah, let me tell you this first. Southern blot, DNA. Western blot, protein. Northern blot, RNA. That's an easy way to remember. All right, so let's start with Southern. Has to do with DNA. What do we use it for? Is to find a particular DNA segment among a vast population of DNA, of different DNAs. So let's say you have a lot of DNA and you want to find if your sample contains a particular DNA segment. What do? You cut down your DNA into fragments using restriction enzymes. You run your DNA on the gel, and then after you have the gel, you're going to transfer. And this is pretty cool. You transfer your DNA from the gel to a film, to a little paper film. And then after you have the film, you're going to add a probe for the DNA that you want. You add a particular probe for the DNA that you want to study to see if it is in your sample. And what happens is you look at the final results to see what gives you any signal. So I think I'll have another summarized one where, yes, you start with a sample, DNA sample, right? You extract it, you run it through the gel, and then you take the gel, you transfer a gel into the membrane, and then here, the membrane, we add we incubate the membrane with a probe. And then if you see any signal here, you will say like, oh, this means that my DNA my original sample contains the DNA of interest. And obviously you're going to have different samples, and you're going to say like, oh, this sample has the DNA of interest, this sample doesn't. And again, this is about DNA, and that's Southern blotting. Northern blotting is the same but RNA. So you extract RNA, you run the gel, and then after you do the gel, you transfer to a membrane, and after you transfer to a membrane, you add your probes for RNA. And then after you add your probes for RNA, you visualize it so that you can see which ones have your RNA, which ones don't. So instead of having DNA here, you have RNA here. And even a more common one, this is way more common, it's called Western blot. Western blot is for proteins, but it's the same idea. It's the same idea, right? You start with your sample, you run it through the SDS page, and then after you run it through the SDS page, your sample here, you transfer your sample to a membrane, right? So this is the transfer process. You don't have to know too much details about this, but you transfer your sample. So this is the membrane, and then after your membrane, you're going to add, because the Western blot is an immuno technique versus northern and southern blot. Why? Again, remember, immuno means antibody. So you use antibodies here, right? You use antibodies, and not only that, we use a primary antibody and secondary antibody. And then from there, you can add something that will visualize it, and in the end you're going to end up with something like this that's going to show you which ones of your samples are expressing your protein of interest. All right. Now, I did a lot of Western blots, and I have published results for some of them. And it might seem simple, but this process takes more than 1 day. Takes more than 1 day. It's very rigorous, but in the end, when you get some really good results, it's well worth it. But this is Western blot. Western blot protein, Southern blot DNA, Northern blot RNA, but same idea. All right. Let's talk about mutagenesis. So, mutagenesis is just generating a mutation. That's what it is. It's generating a mutation. And in vitro mutagenesis, you guys know the difference between in vitro and in vivo, right? In vitro mutagenesis is generating a mutation in a test tube. And we do that a lot. We talked about how you can generate, you can make E. coli cells produce any DNA you want. Well, guess what? You can just go into the code. Instead of giving it a real normal DNA, you can give it a mutated DNA. And guess what? It's going to multiply that mutated DNA, and it's going to make a mutated protein. So I made different mutated versions of my particular protein, and guess what? These mutations were modeling mutated proteins in the disease states. So I remember one of my my proteins has a mutation on position 12. G is replaced by C. But anyway, you do the mutation, you insert it. So I think I have an example, right? So you do the mutation, you're going to insert to do the PCR with the mutation, and then you can insert it into E. coli cells, and the E. coli cells are going to make it. But it's the same that we have here, where whenever you're making your vector, you just add a mutated vector, and the cells are going to make it. Now, one thing that I want to mention that I do a lot, we call it site-directed mutagenesis. Site-directed mutagenesis. You hear this a lot, and it's pretty much a mutagenesis of a particular DNA. You go in and you change one, right? You change one DNA, one base pair, and then you change one amino acid. It's really cool. All right, RNA interference. So you hear people say RNAi, which is RNA interference. Hold on, give me a minute. I want to fix this once and for all. There you go. All right, RNAi is RNA interference, and this is a way to knock down a gene, right? So it's a method to inactivate a gene, right? And it leads to targeted destruction of a selected gene's transcript, and the consequences result in loss of gene function, right? Now, the reason why RNA interference is important is because at first we would use knockout, gene knockout. So when I was taking a genetics class, we learned about knockout mice. But the problem with knockout is you knock out an entire gene, and it causes a lot of problems. So we came up with RNA interference, where you only knock out the gene whenever you target, right? So that means that you're not knocking out all the genes in the body, right? So how does RNA interference work? RNA interference, and again, instead of knockout, we call it knockdown. This was taken from knockout, so knockdown, which down regulating, knockdown, inactivation, things like that. So how does that work? Here's how it works. You start with your double-stranded DNA, right? And we have something called a dicer, right? A dicer is this protein complex that does this, right? So this dicer is the one that's going to come in and open up, right? It's going to open up your DNA, and then a guide strand is delivered, right? A guide strand is delivered there. So the guide strand is called siRNA. So if you've heard of siRNA, this is where it comes in, right? So the dicer opens up your your DNA, and then siRNA comes in and it's complementary to your DNA, right? And then after that, you have this, what we call a risk complex, right? Is the one that comes in and breaks it down pretty much. So you have a dicer, you have a RISC complex, you have guide RNA, you have a siRNA, and then you have the double-stranded RNA that you start with. So this is how we knock down a gene using RNA interference. So RNAi, now you know what it is. siRNA, now you know what it is. And when we get to talk about CRISPR, we're going to talk about shRNA. But I want you to know the difference between knockdown and knockout. And then I want you to pay attention to this method so that you see the difference between this method and CRISPR. Because CRISPR is another version of DNA knockdown. But before we do that, let's talk about high throughput. High throughput technology, when you hear that, is just know that we're talking about automated processes. So all the processes that we just talked about when it comes to DNA, to RNA, to proteins, high throughput methods are just large scale industrial automated methods for that. So you have high throughput sequencing, high throughput expression, high throughput drug discovery, and all that stuff. So this is an example for DNA, but we have many other ones, right? Many other ones that use high throughput technology. So, they use different techniques, but it's the same idea of what we just talked about, but they're using robots, they're using automation, they're using many different techniques. So I wanted to add this section because after everything that we just talked about, people started to be very excited because they say, oh, wow, gene therapy is going to change everything. Because if you can knock out or knock down a gene, then why haven't we not cured all the diseases? Because we know that a lot of diseases are genetic. We know the genes that are responsible. Why not go ahead and knock them out and turn them off? And you have different ways to do that, right? This is an example of using viruses, right? Viruses is actually, this is one of the oldest ways to knock down and knock out a gene, right? Using retroviruses. But we can also use, again, knockdown. You can use those siRNA. There are things that we call miRNA as well. Why are we not using them? Well, the answer is, first of all, some people are using them. We have a lot of clinical trials. We have a lot of drugs that are using this gene therapy technology. We already have some that are doing that, but second, it's not as easy. First, like I was saying, you people were excited because of knockout. Every time we've knocked out a gene, we've had problems. I remember there were animals that I was working with. I was looking at developmental neuroscience, but this gene is called neurogenin. Whenever we would knock it down in a mouse, the the babies will be born, but they will not be past days. They will die pretty much right after being born. So it was a lethal mutation. But even if it's not lethal, knocking down a protein will cause problems. Knocking out a protein will cause problems. But knocking it down is easier. But even easier, for example, I work with drug discovery where you can study the mutation, and instead of using that, you can find a small molecule, a small organic molecule that's going to inhibit the mutation a little bit so that you can knock it down that way. But there are a lot of people working to cure all of the diseases, and this comes on top of gene therapy. Personalized medicine, you can look at somebody's DNA, you can look at what's wrong with the DNA, and then you can personalize the medicine based on the DNA. This is already here because now we can pretty much measure the whole human genome. So I can take your DNA sample, can look at everything in your DNA, and then can say like, oh, okay, you've been having trouble sleeping because you have this mutation. You have a chance of maybe having Alzheimer's, when you hit 70 because of this and that, and this is what you can do about it. So it's not just gene therapy, but personalized medicine is also going to be the future because of all of these technologies that we just talked about. DNA as a code, people are using now DNA as a code because since we know that DNA is kind of like software, right? I remember before I came here, actually, there was a company that talked to me. I had an interview with them. They were recruiting and they were pretty much trying to do that using DNA as a storage system because DNA lasts a long time and is resistant to pretty much corruption. So they wanted to use DNA and build a sequence. And as if you know anything about computers, your picture, this video, everything is just ones and zeros. It's sequences of ones and zeros. And guess what? DNA, you have 4 numbers. Instead of just having 0 and 1, now you have 4. So you can store a lot of information with DNA. But not only that, you can use it to do computation stuff as well. It's really, really cool. And this has pretty much everything we talked about has given birth to a field called synthetic biology, where people take advantage of all of these things to come up with some really cool stuff with DNA. So people have made new types of rice, for example, that's resistant to drought. Right? People have came up with some DNA and RNA. You know that, for example, that's how we got the vaccine. For example, the vaccine is an RNA vaccine. That's synthetic biology. So people have made some really cool stuff using the knowledge about RNA and DNA and the technology that we just talked about. This is another example of making enzymes that can degrade waste. For example, there are people working on making enzymes that can degrade plastics. So we have a lot of technology based on synthetic biology, and they the possibilities are pretty much limitless. CRISPR. Now, you've heard of CRISPR. Recently, actually, I think last year, the ladies who came up with it won a Nobel Prize. And these are two different ladies in two different continents, Dr. Jennifer Doudna, and I think her name is Dr. Emmanuel Le Chantelier. I don't remember, but she's from Germany. But they came up with CRISPR, and this is a very beautiful and elegant way to knock down DNA that is very efficient, have very few errors, very specific. So all of these other methods that I talked about when it comes to knocking down or knocking out DNA. So I talked about knockout, I talked about mRNA, sRNA, RNAi or RNA interference. This one is much more specific. So they're saying that this is one of the best human discoveries since computers, because it has the potential to not only cure all diseases, but people are starting to think of just changing other things other than diseases. If there's a gene that makes somebody tall or makes them have blue eyes, why not do that? So you technically can do that with CRISPR, which is really important. And not only can you knock down a gene, you can also replace it with another gene. So you don't only have to remove mutations or whatever, you can replace the gene with another gene, and it's going to, so you can remove a gene, insert a gene, or replace a gene, which is really cool. So how does it work? It works by using, it actually uses a Cas9 protein, and this Cas9 protein, it's an endonuclease, right? It's an endonuclease, meaning it cuts DNA. And this is a natural protein. So we talked about how, for example, we've been taking advantage of all of this DNA machinery to solve the problems that we have. So this is another example. The Cas9 protein was discovered, I think it's from bacteria, right? It's a defense mechanism in bacteria, but they were like, wait a minute, this defense mechanism is very specific to a certain, to a specific gene. Why don't we use this to our advantage? So that's where it came from, the Cas9 protein. But this is what you do. Right? So this is your DNA of interest, right? And you want to insert a certain gene or a DNA, right? So you have what we call a guide RNA. So you have the Cas9 protein in white with the guide RNA in purple. And this guide RNA is the key, right? The guide RNA is the one that you change to your sequence, right? This is the RNA that's going to guide you into, to your particular sequence. And this one, by the way, is what we call shRNA. I don't see it here, but we call this hsRNA. So the complex, the Cas9 with the guide RNA, is going to come and attach to a matching genomic DNA sequence, right? So we're coming in here with our guide RNA, we bind to the DNA, and what happens? Cas9 and RNA complex is going to cut it. So after, remember the Cas9 is an endonuclease, right? So it's going to cut the DNA. And then after you cut the DNA, again, you can do many things. So while the Cas9 is bound, you can do many things here, right? You can either insert the DNA. So this example is we inserting the DNA where we cut. So you can cut remove something, add something else, or change something else, right? So this is in a nutshell how the Cas9 system works. All right. So again, it's guided by RNA, right? And we call it the guide RNA. And it's from Aspergillus. I think this is bacteria. And since we can make a guide RNA of any nucleotide, any DNA sequence can be edited. Any DNA sequence, right? You just have to express a guide RNA with the same sequence, and then you add the Cas9, and then we can go ahead and target it. And again, CRISPR-Cas9 has revolutionized the genome engineering field, and a lot of people are looking at it to see all of these life-changing things that CRISPR-Cas9 is doing. Oh, by the way, I think I saw this today. Actually, I saw this today that they've successfully used CRISPR to treat cancer. I'm pretty sure that I saw it today, so it must be like either this week or last week where CRISPR has shown to treat cancer. But it can be used to treat a lot of other things as well. And people have successfully done it. And it's just only a matter of time. There's a lot of regulations happening around CRISPR because, again, people can go crazy with it. People can go crazy and try to make different things, but just, it's not over. The story of CRISPR is not over, and it's going to revolutionize medicine. And not only that, since CRISPR was made, people have been actually finding ways to make it even better, make it even more specific, and they have come up with different variants of CRISPR that are even more efficient. So it's a very good method. It's a very efficient method, but I think that's enough. That's enough about DNA and RNA. There's so much we can talk about, but I hope I've, with this video, shown you how it's a whole world and a whole range of possibilities of what you can do with DNA, and you should have an idea of the theory behind the way we do these things, and then the main methods that we use and the rationale behind them. All right. See you later. Bye.